This lecture is based on the second step of how the genes work, and this second step is called as translation. Just like you have a code that has to be eventually understood to follow directions, similarly in the second step of the gene expression is the actual synthesis of proteins. So the actual synthesis of protein is going to take place in this lecture. We pretty much in the previous lecture saw how transcription was taking place, how the DNA was sending its code through the messenger RNA, and recall that all of that was going on in the cytoplasm. This process, however, will take place in the ribosomes. Before we go on to the actual site of translation, let's look at some of the key players in this process. Translation, as the terminology explains, it actually is the conversion of the code to the polypeptide. Recall, the polypeptide is the protein. So the code is coming from the DNA. DNA sent the code through the messenger RNA, and now it's going to be directing the synthesis of proteins. The previous stage was taking place in the nucleus for the eukaryotes, and for the prokaryotes it was actually in the nucleoid region, which is within the cytoplasm. However, translation is clearly going to take place in the cytoplasm. So once the mRNA is manufactured and it is processed and it is mature, remember how we got rid of all the introns, and the mature RNA with its poly A tail and the 5' cap of the G's is mature and coming into the cytoplasm. Now within the cytoplasm, it actually gets in touch with three main players of this process. The first player is called as the tRNA. Recall, the T stands for the word transfer. Transfer RNA is a ribonucleic acid which has a very unique shape, which is sort of like a T. So here is, sometimes in simplicity we will draw it in this format. So over here at one end of this loop are three nucleotides, and these three nucleotides constitute what is called as the anticodon. So the anticodon is going to be over here. Now the anticodons are sequences that will match the codon sequences that are brought by the mRNA. Remember when we had the mRNA was making its complementary copy from the code, and I told you that this is called as the codon. So these codons are going to match the anticodons, which are like a three-pronged nucleotide sticking out of the tRNA. Now on one end it has the anticodon, and on the other hand the tRNA has an amino acid. Now since there are 20 different amino acids, so there are going to be different amino acids pertaining to each tRNA. So each tRNA will have its anticodon and a corresponding amino acid next to it. mRNA was synthesized within the nucleus, and it was a single strand of a chain. So here we have a sort of like a matching between the codon and the anticodon, the codon from the mRNA to the anticodon of the tRNA. And the last player of this whole process is the ribosomes. Ribosomes are the actual protein manufacturing factory, as you can call it. It's the actual place or the actual site where proteins are going to be synthesized. Now ribosomes are actually composed of two subunits, which we will see later in the next slide. Over here I want you to point down the actual structure of the ribonucleic acid, that it actually has an amino acid on one end and the anticodon on the other end. And these are all the different nucleotides that it is composed of. So this is more like the actual structure of the tRNA. Now let's look at the ribosomes and see what the ribosome is composed of. Ribosomes are made up of two parts, and the two parts are sort of like one on top of each other. So you've got one part on top of the other, and it is called as the two parts together are the ribosomes. Now ribosomes in eukaryotes are composed of two subunits, that is 60S and 40S. The larger unit is the 60S unit, and the smaller one is the 40S unit. In the prokaryotes, however, you have the smaller, which is 30, and the larger, which is 50. Now let me just tell you, S does not stand for, like, we cannot just add them up and say it's 100 subunit eukaryote. What S stands for is the word sediment. If any kind of cellular composition is taken into a centrifuge and you spin it, which we did earlier in the semester lab in which we use a centrifuge, then the subunits will sediment at certain levels. So just like a thick broth that you may have and a thin broth, if you were to spin it, and it would lay down the content at different levels. Same thing is with the ribosomes. This is actually a very important factor for drug manufacturers, because when drugs are manufactured, most of the times the drugs are targeted to destroy the ribosomes of the prokaryotes, which is the bacteria. And because if it was to destroy the ribosomes of the host cells, then our ribosomes will also get destroyed by those antibiotics. So this plays an important role in drug manufacture. Now the ribosomes are most of the time are many times put together in a sort of like a bundle. So for example, as soon as one of the ribosome is manufactured by the nucleoplasm and attaches to the eventually when it comes to the ribosomes are actually synthesized by the nucleoplasm within the nucleus. So when they come out and they adhere to the mRNA and they start moving along, so the mRNA does its parts, and then another ribosome attaches to the mRNA. Therefore, eventually on one particular strand, there are many ribosomes that are attached to the same mRNA. And the entire complex is then called as the polyribosomes. So you can see that each ribosome is actually initiating a synthesis of a polypeptide. So you can have numerous copies of a polypeptide being synthesized because they're polyribosomes all working on the same mRNA strand. Pretty much think of the mRNA strand as a string in which is sort of moving along these beaded-like structure, which are the ribosomes. And as it's moving along, it actually increases the polypeptide and its length. So we will go on to some more steps in this process. There are three main steps of translation. The first step is called as initiation, and then is elongation, and then the last step is called as termination. So I'm going to go over these in this section. Initiation, as the name tells you, is the first step. This is the first step, which is initiation, which means that all the translation components are put together, and the proteins, which are some initiation factors, are required to assemble the small ribosomal subunit and the mRNA and the initiated transfer RNA and the large subunit for the protein synthesis. So in the first step, what is happening is that once the mRNA that has processed from the nucleus has reached to the cytoplasm, it eventually will start the process of initiation by eventually moving around. As you can see that it's gliding around this ribosome, and when the first codon is read, And this first codon, keep in mind, will have to be the start codon. So once this first codon is read, which is the start codon, another tRNA molecule is going to come around and attach to the codons, because the tRNAs are bringing the anticodon to the codon, and it will slide off. Then the second tRNA molecule comes in and matches the codon to the anticodon, and it brings along a second amino acid. And on one side, the tRNAs are attaching and matching the codons, and the other side, these amino acids are being attached to the right in the right sequence to form a polypeptide. So they're like this whole step is like a 3-step process that's going on. So in initiation, as pretty much what I'm going to mention is the mRNA is going to enter from the entrance site. The polypeptides are going to initiate forming the right amino acid matching the codons, and then it will move on. So this would be the first step, which is initiation. Now in the second step, which would be let's actually look at initiation in the second step, which is the elongation. Now in the elongation process, what is happening is the protein step is actually increasing its length by one amino acid at a time, because the first amino acid has attached. The mRNA glides over, and when it glides over, the second amino acid will come from one side, which is actually, want to bring you attention is there are two things happening simultaneously. One thing is the transfer, the mRNA is sliding, and the secondly is bringing the right amino acid to attach to the polypeptide. So you can see that the polypeptide is actually getting longer as it's gliding over. So one is the A site, which is the arrival site, if you can remember it that way, and E, which is the exit site. So the second tRNA will arrive at the A site, and the P site, which is the site where the polypeptide, which is present, this is the peptide site, will glide over, and then it will shift to open up another A site, and another amino acid is coming in. So you can see that from here we have two anticodons matching the codon, then it glides off, the next one comes in, this is an empty slot, and then this will fit into this slot. The next amino acid is going to be attached, it moves on. Now this is kind of hard to visualize it on one flat surface. I really would like you to watch the animation on this section, because the animation I think will be easier to see, because you can see the amino acid coming, the codon and the anticodon matching, and so forth. Now in the last step, which is called as the termination, once the ribosome reaches the stop code, which is a code on the mRNA, and this code will not code for any amino acid. Remember, we said there was no amino acid for the stop code. So when this reaches the stop code, there is a release factor that attaches, and the subunit will detach, and then the polypeptide is free, the mRNA is free, and it can start all over again. So this concludes our first section, and then we'll start with the next section soon. Now